Hey, good morning, AP Bio folks. Welcome to another little video lecture on the information in Chapter 4. So I just wanted to give you a quick look at Chapter 4, which basically gets into what I like to call organic chemistry or carbon and the molecular diversity of life. Ugh, it's kind of a mouthful. If you go into the folder, there is I call them skeletal notes for Chapter 4. It's page 13. And I'm going to follow along with that. And you guys can follow along as well. Jot down some notes as we go through it. And in the end, you should have a highlight of sort of Chapter 4. And it should hopefully help you out with the reading guide as well. So here we go. Chapter 4. Quick look at organic chemistry. One, it has to do with carbon. That's what the definition of organic chemistry is. It's when it has carbon involved. So when you look at our definitions here, organic chemistry is a branch of chemistry that specializes in the study of carbon compounds. So we're going to talk about all these things that contain carbon, like proteins, fats, lipids, steroids, nucleic acids, sugars, cholesterol, all this stuff, right? Organic compounds are compounds then that contain, obviously, carbon. And they typically have a lot of hydrogen. So there's kind of those hydrocarbons that you guys were looking at. The major elements of life, you guys may remember from biology, we talked about CHNOPS, right? So CHNOPS was carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Those are the major elements of life. They make up the majority. There's trace elements of a bunch of other things off the periodic table, but for what we're going to look at, it's mostly a high percentage of carbon as the base, lots of hydrogen, nitrogens in certain molecules, oxygen, obviously, phosphorus, and sulfur. The thing about carbon, and the thing that makes it so important, is it can form these huge, complex, real diverse molecules that we're going to look at over the next week, going into next week. Specifically, we're going to hang out with proteins quite a bit because they're such a diverse thing that we'll take a look at next week with a little lab on those. But that is sort of our importance of carbon. Now, when or as we look at these different things, hopefully you remember this. If you don't, you can bust out your periodic table. But something about carbon that makes it so cool is that it has 4 valence electrons. Now, if you don't remember what a valence electron is, hopefully you do, but if you don't, the valence electrons are the ones that are in the outermost orbit of the element. So in this case, carbon has 6 total, right? 1, 2 in that first orbit, and then 4 in the second orbit, which makes it super, super capable of bonding to pretty much everything. All right, so it can form up to 4 covalent bonds. And again, if you remember the molecular building lab that I had you guys do the other day or last week, it was the black piece that had 4 little prongs. Those prongs are representing the bonds that it needed to have to make it stable. Its most frequent bonding partners, you can probably take a guess at this. Number 1 is probably hydrogen, right, for those hydrocarbon chains. It also bonds with oxygen a fair amount and with nitrogen. So those are kind of its major bonding partners that it has. Now, continuing on with looking at it, one of the other things we have to know about these covalent bonds is the bonds, and again, this is the way that it becomes stable. The bonds can be a single covalent bond, or a double, or a triple. All right, those are all types of covalent bonds. So if you look at the image that I have here, right here, all of these single lines are single covalent bonds. And guys, you remember that these are a sharing of 2 electrons, whereas here there's a double, so each one is sharing 2, so that's a complete sharing of 4 total. And then here, each one has 3 involved, so we have a sharing of 6 total. All right, so that kind of talks through some of this the structure of the bonds and why carbon is so cool. Carbon can form large molecules. There's four classes of macromolecules that we are going to look at. There are the carbohydrates. Okay, that's one big major macromolecule. There are proteins, which we're going to spend a little bit more time on because they're pretty diverse. We have lipids, which we typically think of as fats, but there are other things in there. There's waxes, there's steroids, there's some other things. And then lastly, we have nucleic acids, and we know that from our genetics and DNA discussions. So, all that being said, carbon is pretty dang amazing because of all of those reasons. It can also form all kinds of crazy shapes. It can form singular branches like this with double or triple bonds. It can form these branched pieces where it's going to have things coming off some central carbons. It can form rings of different shapes, sometimes all singles, sometimes there's double bonds. So it has this ability to form just a multitude of different shaped things, which is pretty important. It also, it forms these things, and this is where the new stuff is going to come in. A lot of that you guys already know, I hope, but it forms these things called isomers. Isomers are molecules that have the same molecular formula, but they differ in the atom arrangement. Now, this actually is kind of cool, and we talked about this a little bit, back in biology, but when you have different structures, they have different properties, all right? And the one example I'm showing you here is what they call a structural isomer versus a cis-trans isomer. And that one you may remember from when we talked about the way different fats are formed. And then the third type, they call them enantiomers. Oops, if I could learn how to spell would probably be great. Enantiomers. All right. When you look at the different things here, structural isomers, if you look at the two differences here, the formula for this one and this one is exactly the same, but they vary in the arrangement of the covalent bonds. So they've covalent, yikes, arrangement. Cis-trans differ in their spatial arrangement. So this one has right here the X and the X, whatever that is, it's on the same side. So this one here is the cis. This one, they are on opposite sides. So that one is the trans, right? And there were different ways of looking at these, and I'll show you a couple examples here. And then the enantiomers are ones that are basically mirror images of each other. So if you look at this one, it has that, and this one is the mirror image. Okay, now why do those matter? Well, they do. They matter quite a bit, actually. In drug production, there's this thing called thalidomide, all right, and it has different enantiomers of it, and the enantiomers cause it to have super different effects on people. The good enantiomer is a drug they use to reduce morning sickness. So people that were pregnant would take this because it would help them with their morning sickness. If you've ever known someone who's pregnant and has morning sickness, it can be pretty nasty. It can be pretty awful. So they would take this. It was marketed back in 1957 in West Germany was the first time they used it. They used it primarily as a sedative, but they also started to realize that they could use it to treat nausea, and it alleviated morning sickness in pregnant women, which again was a good thing. But what they discovered throughout the world, about 10,000 cases were reported of infants that had sort of these birth defects. About 50% of those 10,000 that cases reported survived. And what happened is they had these very shortened limb deformities and things like that. So they also had malformed hearts and eyes, blindness, deafness, just a lot of issues with it. And what happened is the good enantiomer, so the version that they wanted to use originally, it reduced morning sickness. The bad enantiomer, that one would cause birth defects. What would happen is within the patient's body, the good was converted to the bad by the patient's body, and it was causing these birth defects to happen. Right? It's still used today. I did a little research on it, just kind of looking into it. They use it now to treat certain cancers or leprosy, HIV. So typically what would happen was the mother would take it. The mother was fine, but the babies that were born would have it. So they're using it now with people that are basically deciding they're not going to have children because of whatever reason it is. It could be age, it could be this, these diseases or these things that they have are sort of eliminating that possibility in their lives. But the enantiomer, if you look here and here, it's basically that whole mirror image idea, and that would cause those problems. So it's kind of an interesting look at sort of the way that a simple thing, even though it looks pretty much the same, it's flipped, and it would cause major differences. This is showing you some other ones, more common ones. Pharmacological importance of enantiomers. You guys all know ibuprofen, right? Ibuprofen is for pain, inflammation, things like that. The effective enantiomer is this one. The ineffective one is the mirror image of it, and it doesn't work nearly as well. Just different types of ibuprofen. Albuterol, if you guys have asthma or anyone that uses inhalers, those are typically albuterol. It's used for asthma. Again, effective, ineffective. So they have to make sure they're producing the correct kind and not having any issues with that. Just a throwback to what you guys were doing the other day, the models last week. A lot of the functional groups, the reason I introduced that to you was that is part of what makes all of these organic molecules different. They behave differently based on the functional groups that you have. And then, again, you guys built a lot of these with the hydroxyl, carbonyl, carboxyl amino, sulfhydryl, phosphate, and methyl. And again, you're not going to have to draw these out of memory or anything like that. So what I just threw on here, just so you had it, was this was page, if you remember, page 64 in your text, was this document that had the different ones that you'll look at, and you just want to be familiar with these. So hydroxyl, carbonyl, carboxyl, and then the other ones as well. So chapter 4 is fairly quick. Again, it's with a couple additives to it, talking about some of the chemical stuff, but it's a review of chemistry for you again, looking at those functional groups, looking at the connections, and now we can dig into, starting next week, or actually later this week, we'll get into carbs, and then next week we'll spend with proteins mostly, but we'll also touch on lipids and nucleic acids. So that is it. I just threw that last little table on there as well, kind of breaking it down specifically on one page, the functional groups for you. So if you have questions about the chemistry stuff, certainly let me know. Otherwise, go through that chapter 4, scan through there, read it. I am asking you to complete your reading guide and turn that in. It's a little more detailed one than the ones in the past, but that's because the stuff's starting to build up in knowledge a little bit. So there you go. Have a good one, and we will talk to you guys soon. Stay healthy, stay happy.